Hi, I'm Stephanie Schopmeyer, a research scientist at the Florida Fish and Wildlife Research Institute and a member of the Florida Coral Rescue Team. Apologies for not being there in person, but I'm going to present some tips and guidelines for developing a collection and biosecurity plan for healthy corals in response to stony coral tissue loss disease, using criteria developed by the Florida Coral Rescue Team and advisory teams and working groups from the Association of Zoos and Aquariums. As the overall goal of coral rescue is to collect and gene bank healthy corals, which will serve either as the parents of future generations of coral offspring that will be used for restoration, or as the corals themselves, it's important to consider a variety of criteria when developing a collection and biosecurity plan. These criteria include species prioritization, genetic management, health assessment, transport, system biosecurity, and system preparation. The first step in developing the collection plan is to prioritize which coral species will be candidates for rescue. Prioritization might be based on susceptibility to the disease, the speed of disease progression across the colony, the prevalence of whole colony mortality, the contribution of the species to reef building, the current abundance and spatial distribution of each species within your region, the conservation status, or the species reproductive strategy. Other major considerations might also be the known amount of space available for holding or the possibility for expansion, the number of staff available to provide care for the corals, and funding. In Florida, we ranked each species into one of four categories. High priority target, where mostly species of highest concern and susceptibility fell into this category. High priority opportunistic, which were mostly species with high susceptibility, but those that were more rare on the reefs and may only be found in specific habitats and therefore may not be found at all collection sites visited. Medium priority, or species with later susceptibility or disease progression, but are important components of reef building. And lower priority, or species with late susceptibility and lower mortality. Our prioritization resulted in 16 high priority species and 4 medium priority species, 20 species in total. Only high and medium priority species were included for collection in Florida due to funding, space, and coral care limitations. Lower priority species remain more prevalent along the Florida Reef Tract, which allowed us for limited resources to be more focused on species with the greatest conservation needs. The second step in developing a collection plan is to consider the potential genetics and the genetic management of your collections. You want to make sure that sufficient genetic diversity will be collected to support restoration activities. The genetic target for restoration, based on guiding principles in common conservation literature, is 50 unique individuals or genets per species. Using this conservative estimate helps avoid inbreeding depression and limits the impact of genetic drift. Here in Florida, without the ability to conduct genetic analysis during collections and actually know how many genets were being collected in real time, we set goals to collect a minimum of 200 individuals per species in hopes that within the 200 colonies, we would have 50 unique genets. This became more difficult as the disease margin progressed, as we were trying to collect at least 5 to 10 miles ahead of the disease margin to reduce the risk that the corals had been exposed to stony coral tissue loss disease. To increase diversity, collecting a limited number of each species from each reef, for example, we only collected 8 colonies of each species from each reef, will assist in maximizing genomic diversity by collecting from spatially and geographically different populations. In addition, this will maximize the diversity of breeding species as their dispersal distance is lower. You can also increase the odds of collecting genetically diverse corals or corals with potential adaptive qualities by collecting from different habitat types and depths within a species range. The Florida Genetic Management Plan includes the development of genetic markers for all 20 rescue coral species so that all rescue corals can be genotyped to ensure that 50 unique individuals were collected. To date, markers and genotyping have been completed for 3 species: Diploria labyrinthiformes, Pseudodiploria strigosa, and Meandrina meandrites. So far, we have met or exceeded our collection goal of 50 unique genets. The genetic markers for all 20 Florida rescue species will be made publicly available. Please see Alicia Vollmer's talk on Wednesday at 3:45 for more information. Finally, rescue projects and programs should utilize genetic catalogs or databases to house all of the genetic information collected during coral rescue activities and make it available to the broader restoration community. Step 3 is to determine how corals will be selected from the reef for inclusion within rescue holding. As these corals will serve as the brood stock for future generations and for the majority of corals that will repopulate the reefs in response to stony coral tissue loss disease, it's important that the brood stock is as healthy as possible upon collection. First, look at the size of the coral. Most species affected by SCTLD are reproductive at sizes greater than 10 centimeters in max diameter. So collecting corals between 10 and 30 centimeters max diameter will ensure that adult corals are being collected, and that will hopefully produce offspring soon while not being too heavy for divers to carry or will take up too much valuable space in holding tanks on land. Second, look for colonies with live tissue edges that aren't buried in the sediment or have tissue competition with surrounding reef organisms. Third, avoid collecting corals with any type of active tissue loss, including abrasions and other irritations. Avoid corals with any type of Cleona sponge infestation or displaying signs of bleaching, paling, or discoloration. In addition, avoid colonies with multiple boring organisms such as polychaetes or bivalves, and do not collect corals with greater than 10% old mortality. In other words, look for the healthiest possible corals. Look to see how easy it will be to remove the colony from the substrate. If the tissue is damaged during collection, this may affect their overall health. As fragging creates more corals or clones, this does not necessarily increase their reproductive potential or genetic diversity, and some corals may have difficulty fusing back together. If any part of a colony does not meet the health criteria, no part of the colony should be collected, as some diseases are systemic and may just not be visible on the surface yet. Step 4 is to figure out how to best transport the corals between the collection site and the land-based facility to keep the corals healthy, in good condition, and stress-free. When transporting corals, ensure that the conditions are and remain to as close to the conditions that were present at the collection site. This will help prevent stress to the corals. Use clean, insulated containers, which will help maintain temperature. In addition, keep the corals out of the sun, even if the containers have a lid, as this will also help maintain water temperature. If transporting corals in water, fill containers to the top to prevent the corals from sloshing around and prevent creating abrasions on the corals. We've even used bubble wrap to make little bumpers between colonies, but make sure to not damage fragile tissues by tightly wrapping the individual corals. Prevent crowding in containers unless the transport is very short, such as less than 1 hour. For branching species of whole colonies, less than 75% of the total volume of the container should be filled with corals. If fragments of branching species are used, less than 30% of the total volume of the container should be filled with coral. This is the same amount for massive species. If you're using really large transport containers, you may use some type of shelving to help distribute the corals throughout the water column. However, the shelving and coral should not fill more than 30% of the total volume of the container. Circulate and aerate the water with pumps and large air stones for any transport greater than 1 hour. Dissolved oxygen levels should be at 4 milligrams per liter or higher. Water pumps and air bubblers should not be directed directly at the colonies, as this may damage the tissues. Maintain water temperatures within 1 to 2 degrees Celsius of the collection temperatures, but never exceed 30 degrees Celsius. Also maintain salinity to the collection part per thousand. You should conduct water changes every 2 hours during transport, and each water change should replace 50% of the water volume of the container. You should use artificial seawater or water from the collection site, but never use water from marinas, intercoastal waterways, or untreated canals or wells. Do not transport for longer than 8 hours at a time. If your transport is going to be longer than 8 hours, arrangements should be made for overnight storage in a flow-through system. Upon arriving at your destination, all corals should be visually inspected for evidence of transport stress, such as bleaching or paling, any type of tissue loss, changes in tissue condition, such as swelling or thinning, or excessive mucus production. To further protect your corals, corals must not come in contact with pathogens or organisms that originate outside of your local region, nor water, equipment, food, or anything that might carry stony coral tissue loss disease or other pathogens. Therefore, strict biosecurity guidelines should be followed. Here are some of the guidelines to consider. Artificial or natural seawater should be used, and natural seawater must be treated using ozone dosing or UV exposure. Rescue corals should be held in separate systems and even different rooms if possible from other systems holding corals, especially if those systems are holding non-endemics. For example, tanks holding Florida corals should not be in the same room as tanks holding Pacific species, or corals exposed to SCTLD should not be held in the same room as pre-invasion Caribbean corals. If systems cannot be separated by rooms or a sufficient distance, splash guards should be placed around each tank to prevent splashing. Only use cultured live rock or biomedia acquired from the similar collection region as the corals. For example, all systems for Florida coral rescue used live rock collected from Florida or biomedia seeded with Florida live rock. Additionally, pre-invasion corals are only held with pre-invasion live rock and not rock that has been exposed to SCTLD. Do not use live bacteria starter products or live sand. All co-inhabitants should originate from local sources or from similar collection regions as the corals, and all live rock and co-inhabitants should be quarantined for at least 30 days. Frozen food or commercially processed dry food, such as pellets, flakes, or powders, are acceptable, but live cultured food should be cultured in isolated systems to avoid cross-contamination, especially those holding non-endemics. If buying food from outside sources, adequately vet the vendor to make sure the food is locally sourced for your region. Conduct activities to avoid cross-contamination. Don't go back and forth between project tanks unless strict disinfection protocols are followed. These might include disinfecting your hands and arms with soap and 70% isopropyl alcohol, and changing your gloves and soiled or wet clothing between systems. Projects involving rescue corals should have their own dedicated, clearly labeled equipment for use in project systems, including things such as scrub brushes, siphon hoses, and viewing boxes. These types of equipment should be cleaned using strict disinfection protocols. Post signs to alert staff of additional biosecurity requirements related to SCTLD and limit project access to those trained in these biosecurity requirements. Visitors should be prevented from coming into contact with the project systems, including not touching the tank structures, the water, and especially the corals themselves. The final step in the process is to ensure that the system in which you're holding your rescue corals is properly sanitized and disinfected. Systems with any reused components must be sanitized and disinfected. If the system is not 100% new, this includes all piping and life support system components, then it must be sanitized and disinfected. All surfaces should be free of biofilms. To remove residual organic material, the system should be manually scrubbed and rinsed with fresh water to remove algae, detritus, and calcified material. Remove any and all biomedia, live rock, and substrates from previously used systems. Only new biomedia and live rock should be used. Next, the system should be disinfected using a free chlorine solution concentration of 200 parts per million. This means that all parts and equipment should be soaked in, or a free chlorine solution should be circulated through the entire life support system for a minimum of 4 hours. The chlorine should then be neutralized using sodium thiosulfate and rinsed with fresh water. Finally, the system should be disinfected with 2 rounds of peroxyacetic acid, or PAA, at 15 parts per million for 6 hours. If you have any questions or would like to discuss anything that I've presented here, please feel free to reach out through email. Thank you and enjoy the rest of the workshop and ReFutures conference.